Hello everyone, I am Dr. Soumya Das, Associate Professor, Noida Institute of Engineering and Technology, Greater Noida. So, as we already know, we are studying about the human anatomy and physiology. Today, I am starting your Unit 2, Lecture 7. The title of today's class is Organization of Skeletal Muscles, Physiology of Muscle Contraction and little bit small introductory class of neuromuscular junction along with the role of various neurotransmitters. Let us start. Starting with the introduction, you will see how the skeletal muscle look like, right, the diagram in the right hand side. So, starting, skeletal muscles are the muscles which are attached to the skeleton. It overall carries 40% of human body mass. Skeletal muscles mainly used or function for locomotion and it is showing voluntary relaxation. So, students, do you remember what is the meaning of voluntary action? We already studied in our previous lectures. There are two types of actions, one is voluntary and the second one is involuntary actions. Voluntary means according to our will, which we can control by ourselves, that is the rotation of hands, which includes the skeletal muscles, and blinking of eyelid is coming in involuntary action, heart beating involuntary action. Okay. So, those actions which we can perform as we required, that is coming under voluntary action. So, here we are talking about the skeletal muscles. Skeletal muscles basically covers all the bones and skeletal system. So, the actions which needed in our locomotion, sometimes change in position and during exercise, sports, we need the voluntary role of skeletal muscles and basically here is the relaxation action. Like nervous tissue, the skeletal muscles are excitable in nature. What does it mean? Excitation means the skeletal muscles showing or having a great ability to respond towards a stimuli. When our nervous system gives any stimulus to the skeletal muscles specifically, it will relax or it will respond specifically. Unlike nervous tissue, skeletal muscles are having three main characteristic features, which is enlisted below this. The first nature is contractibility. It means the muscle, they are contractile in nature. During the time gap, it may vary in their length. When contraction, it may become short. During relaxation, skeletal muscle relaxation, it may become long in length. The second characteristic feature of skeletal muscles is extensible. What does it mean? It means they can extend or stretch when required. And the third most important one is the elastic nature of skeletal muscles. It means the muscles, they can return to their original shape, elastic, just like you see the rubber band, the nature of the rubber band, how it becomes contracted and relaxed in long length. So, it means it is highly elastic in nature. Now, coming to the arrangement of skeletal muscles, how the different layers will arrange one over the another. So, a single muscle cell, we are starting from the basic cells, after that we are going to the tissues and then in a particular organ. So, a single skeletal muscle cell is also considered as a muscular fiber. So, there are several fibers which made up of a structure named myofibrils, right. What does this myofibrils do? Myofibrils are made up of thick as well as thin filaments, which we have already studied in our unit 1 lectures in the sliding filament theory. Do anyone remember? Okay, very good. So, myofibrils are made up of thin as well as thick filaments and this form different striations. Striations means student band like characteristic feature. Striations of dark and light bands. Beneath the myofibrils there is a structure named sarcolemma. Sarcolemma is the skeletal muscular cell membrane. Inside the membrane, sarcoplasm, muscle cell and cytoplasm is present. The length of each myofibril, which is a fiber like structure, myofibril is divided into repeating units, which is named as sarcomeres. Okay? So, a single sarcomere, if we are talking about, is considered to be as a functional unit of a skeletal muscles. So, always remember the functional unit of a skeletal muscle named is sarcomere. Muscles consist of so many packages of muscle cell which is known as fascicles. Okay? The skeletal muscles are having layers or we can say bundles of muscular cells which is fascicles. A muscle cell is long and spindle in shape. Coming to the physiology of skeletal muscle contraction, or this term we can also say about the sliding filament theory or the mechanism of skeletal muscle contraction. Okay. So, see the diagram students, what you are observing here, the red thick band is made up of myosin filament, while the upper one, the parallel one just above the red thick band, that is myosin filament, you will here see the yellow thin filament made up of actin protein. So, there are basically two proteins, one is actin and the second one is myosin. Actin is considered as thin filament, myosin is a thick filament. Thick means it is making a broad layer of the skeletal muscles. So, several parts are here. Let us see one by one. You will see in the actin thin filament, here one protein troponin T is there. Troponin are found in three different types. One is troponin T, troponin C and troponin I. You will see in the diagram clearly here. It is showed in different colored structural spherical balls. Then the complete yellow one is the thin filament which is actin, made up of actin protein. After this you will see a dark black band straight line, that is alpha tropomyosin. It is also a protein which covers this thin filament. After this there is a cross linked bridge which is myosin binding protein C. Okay, this one myosin binding protein C. So, this is complete your thin filament made up of actin protein, and beneath this there is beta myosin heavy chain. Okay, this is beta myosin heavy chain, and after this students we are having thick filament, the red thick band, that is made up of myosin protein. Apart from myosin, we are also having here another protein named titin. Okay, and this is basically essential myosin light chain, okay, which is also termed as MLCK protein students. What is the full form of MLCK? Myosin light chain kinase protein, C MLCK, that is myosin light chain kinase, right. Kinase means it is a protein required for the sliding filament mechanism of myosin thick band. Okay. So, beneath the titin there is essential myosin light chain which regulate the myosin light chain to get overlapped with the thin filament which is made up of actin protein. Now, coming to the text part, students, what exactly the sliding filament mechanism is. So, the length of complete skeletal muscle get shortens during contraction or when we are doing any physical work, okay, contraction. Why this occurs? This occurs because of the presence of thick and thin filaments. Thick is, always remember, thick filament is made up of myosin protein, thin filament is made up of actin protein. So, the reason, because the thick and the thin filaments slides over one above. Okay. So, this is sliding filament theory and this process is required for the skeletal muscle contraction or relaxation. When the both the layers, thick and thin filaments, are apart from each other, it will means the skeletal muscles get relaxed now this time, and when they are cross bridging, or we can say overlap one over the another, it means there is a physiological change of skeletal muscles contraction. Now, at this time our skeletal muscles or our body is doing any specific action. The thick filament consists of 300 myosin molecules. Okay. So, the myosin, which is the thick filament, composed of two parts. The first one is the myosin tail, named as myosin tail, the tail structure, and the second one is the myosin, that is, you will see here, this is myosin head, students, and the composed detailed longitudinal long structure is the myosin tail, and the spherical one is the myosin head. Myosin tail forms the shaft of the thick filament and the head projects towards the thin filament. The thin filament, which is the actin protein, it is composed of three proteins basically. The major is the actin protein. Apart from actin, the thin filament is having troponin and tropomyosin proteins. Here in the diagram we already see the troponin. Troponin protein is found in three forms, that is T, C and I. Apart from troponin protein, tropomyosin and actin proteins, all three forms of protein make the thin filament, which is very much required for the sliding filament mechanism of the skeletal muscle contraction. Okay. Myosin tail, coming to its working part, the myosin tail forms the shaft, means the complete rest body part, shaft of the thick filament, while the myosin head projects towards the thin actin filament. Thin filament consists of actin, troponin and tropomyosin proteins. At the onset of contraction, what does it mean? Onset of contraction means when our body, when we do any physical exercise, physical change in our body posture. So, the onset of contraction when start, the sarcoplasmic reticulum releases calcium ions. Okay? जो आपके sarcoplasmic reticulum cytosol में present हैं, they will release so many Ca2 plus ions where in the cytosol, that is the fluid medium. Then the release of this excessive calcium ions, we already know the calcium ions are responsible for making contraction. Okay. So, this excess release influx of calcium ions inside the cytosol is responsible to start to initiate the action of skeletal muscle contraction. There they bind to the excess of calcium ion. Now, where it goes? It goes and binds to the troponin protein and causes troponin tropomyosin complex. In the presence of calcium ions inside the cytosol, the troponin protein binds to the tropomyosin and it makes a complex molecule, and this complex now moves away from the binding site of the thin filament, which is made up of actin protein. Once the binding site is free, okay, this complex moiety, when it gets free, the repeating sequence of the events of skeletal muscle contraction cycles start, and this finally causes the thick and thin filament to slide on each other. Okay? And this is the start of the skeletal muscle contraction phenomena. Now, there are basically 4 steps which are involved in the skeletal muscle contraction. The skeletal muscle contraction cycle comprises of basically the following 4 subparts. First one is the which is very much required, or we can say important for the start of contraction, skeletal muscle contraction, that is the ATP hydrolysis. I hope you already know about the ATP. So, the full form of ATP is adenosine triphosphate. What does this ATP do here? Okay, very good. So, the use of the need of ATP here to get energy, okay, to start up of any action. What action we required here? That is the skeletal muscle contraction. So, in the first step we need the energy, source of energy is the ATP here. So, hydrolysis means in the presence of water, when this ATP molecule gets break down, so there is attachment of the thick myosin protein with the ADP and PI. PI here is the phosphate molecules. It means the moieties get activated to start the particular function. After ATP hydrolysis, the second step is the attachment of myosin, that is the thick filament, to the actin thin filament, and this attachment makes a cross bridging, students. Okay, just a bridge like structure. After this, there is a need of power stroke. So, we have to know about what does this step doing. The power stroke is nothing but just the what is happening here? The myosin cross bridges rotate, just they change their configurational structure, rotate towards the center of the sarcomere, and this generate the startup of the power energy, okay, power stroke. And the last step is the detachment of myosin filament from the actin filament. So, coming to the structure, students, little bit in detail, we have to study the key role of initiating of this skeletal muscle contraction is the release of calcium ions inside the cytosol, which is very much important. Okay. In the first step, we will see here myosin head, right, this is the myosin head. Head first get hydrolyzed in the presence of adenosine triphosphate, that is ATP, and this becomes reoriented and energized because it get the ATP structure. So, it gets energized, and after energizing this ATP molecule, students, after hydrolysis will convert into ADP phosphate. It means adenosine diphosphate, and P structure you will see here is phosphorylation. So, for startup of any action, the phosphorylation part is required and must. So, this is the first step. In the second step, you will see here, after phosphorylation, the myosin head is activated. Activated means it is now ready to make a cross bridge with the thin actin filament moiety. Myosin head binds to the actin, forming the cross bridging. Okay, you will see here, this makes a good cross bridging structure. After this, the ADPs get released, and then the third step, which is known as the power stroke of the contraction of skeletal muscle, myosin cross bridges rotate, change their configurational structures, and it goes towards the center of the sarcomere, and this step is known as the power stroke. Okay. After this third step, the ATP molecule, the new ATP molecule is present, and it is ready for binding with the new fresh moiety of myosin thick filament. Fourth step, as myosin head binds with this new ATP molecule, the cross bridging between the actin and the myosin filament gets detached from the actin part. Okay. It means this step is terminated. Okay. So, you will see the fourth part is the detachment of myosin from the actin. The myosin head bind with the ATP, fresh ATP molecule, and the cross bridging is stopped this time. Okay. So, it means the skeletal muscles which is earlier contracted now in a condition to get relaxed. After again the new action, when the start of new action, new physiological change when required by skeletal muscles, the same steps will start in the sequential manner. So, starting in short, just the brief about the skeletal muscle contraction, or we can say the sliding filament mechanism. The first one step is the ATP hydrolysis. The myosin head, which includes the ATP binding site, and here the involvement of ATPase. ATPase is the students, ASE means enzymes which is required to break down this ATP molecule. This ATPase enzyme are required for hydrolysis of ATP, adenosine triphosphate, to the adenosine diphosphate plus 1 phosphate group will be released. And this hydrolysis with the help of ATPase moiety gives energy to the myosin thick filament myosin head. Okay. So, ADP and a phosphate group which is now remain attached to the myosin head in the first step, means in case of ATP hydrolysis when the first step occur. In the second stage, which is the attachment of myosin to the actin and making a cross bridging, means the overlapping of thick and thin filament. The energized myosin head here attaches to the myosin binding site which is present at the actin protein site and releases the previously hydrolyzed phosphate group. When the myosin head attaches to the actin protein during the contraction, they are referred as a cross bridging. Why? Because you will see here a cross bridge like structure is formed. Above there is the actin thin filament and below, just beneath this layer, is the thick myosin layer. In the third step, that is the power stroke, once the cross bridging occurs, the power stroke step is initiated. What occurs in this power stroke? This cross bridge change their rotation, and this rotated part, complex moiety, goes towards the center of the sarcomere, and here again they release the adenosine diphosphate molecule. The cross bridges generates a force which is known as the power stroke, a force which finally slides the thin filament over the thick filament, right. Just you have seen beneath the thick filament there is a thin filament. So, a force or a energy is required which slide or make the cross bridging like structure. Now, at the last fourth step, detachment of myosin from the actin moiety. At the end of the power stroke, the cross bridge complex structure remains firmly attached to the actin protein until it binds with the another molecule of fresh ATP. Okay. So, when there is fresh ATP they will found near their surrounding, the fresh ATP then binds to the ATP binding site of the myosin thick filament head. This myosin head detaches at this point of time with the thin actin filament. So, the cross bridging is now get separated. It means our contraction, skeletal muscle contraction now step is ends and there is start of contraction, skeletal muscles relaxation. So, in the table you will see there are so many types of muscle contraction. First row you will see features, after this the isotonic nature of the skeletal muscle, and in the last there is isometric nature. So, the length of skeletal muscles, in case of isotonic it will change. Iso means same, tonic means students tension, pressure, okay. Or in case of isometric, iso means same, metric is the meter unit of length. So, what is same here? The length of the skeletal muscles is remains same. Now, coming to the tension, in case of isotonic, iso means same, tonicity means tension. So, it is the tension of the skeletal muscles is same, while the isometric changes. Movement of the object occurs with the help of skeletal muscles when it is isotonic, but there is no movement of any object you will see when the isometric startup. Extension of work done. Isotonic, the extension may can extend by using the skeletal muscles, while in case of isometric there is no further extension of work done will be occurred. Examples of isotonic skeletal muscle contraction: lifting weight, walking, running and flexion of arms. Flexion means students when we are doing any work with the help of using skeletal muscles above the force of gravity, means the reduction in the size of angle like this, okay, bending our arm, hand, twisting our leg upside, that is the angle between these two becomes decreasing. This is the flexion. And in case of isometric, the contraction of anti-gravity muscles, like for example, anti-gravity role of skeletal muscles, like when we are jumping, the anti-gravity action or the movement of jaw during talking or chewing is the action which is considered to be as anti-gravity skeletal muscles contraction. When we are changing our posture, okay, or when we push any hard object or we can say a wall, it is coming under isometric change of the skeletal muscle contraction. Now, coming to the neuromuscular junction or we can say motor end plate. So, it is what is neuromuscular NM junction? It is a junction between the motor neuron and a muscle fiber. So, what is happening here? The action potential is the minimum potential or energy which is required to start up of any action. The action potential from nerve is transmitted to a muscle, motor end plate में, muscle fibers तक, through this neuromuscular junction, and the structure consists of presynaptic membrane. After presynaptic membrane we are having synapse or we can say synaptic cleft, and after this postsynaptic membrane. So, see the diagram students, this is the motor nerve and this is the skeletal muscle fiber. So, this is the preganglionic nerve ending, and what you will see here, you will see in the circle there are so many molecules of acetylcholine, that is excitatory neurotransmitter is present here, and beneath this in the muscle fiber this is the receptors which receive this acetylcholine molecule, and this presynaptic nerve ending will join by the release of this neurotransmitter with the postsynaptic neuron, and by this the impulse or the messages transfer from the presynaptic to the postsynaptic neuronal endings. So, the presynaptic terminal is the axonal terminal of a motor neuron. The axonal terminal consists of a number of synaptic vesicles, and these vesicles contains so many different type of neurotransmitters. Just you have seen here, this is acetylcholine neurotransmitter which is released upon receiving a nerve impulse. The synaptic terminal also contains calcium channels, and after fluidization of this calcium channels, there is a release of neurotransmitter acetylcholine in the synapse or we can say synaptic cleft. These channels are voltage gated calcium channels which open when a nerve impulse reaches the presynaptic axonal terminal. Now, coming to the synaptic cleft, it is having 50 to 100 nanometer wide and filled with extracellular fluid. The muscle fiber is covered by a basement membrane of basal lamina, and it contains, you will see here, acetylcholine neurotransmitter, and apart from ACH there is also acetylcholine esterase. ACE means it is enzyme and it is required acetylcholine esterase required to hydrolyze the ACH acetylcholine to acetate and choline moieties. Now, postsynaptic, or we can say the end membrane potential, it is a part of sarcolemma and lies under the terminal button. It is thrown into several folds, or we can say the junctional folds, and so it is required to increase the end plate surface area. And it also contains the acetylcholine receptors, which contains voltage gated sodium channels. And this is the neurotransmitter we already talked about, the acetylcholine. Acetylcholine is a neurotransmitter which is required for the impulse or the message transfer, so that the physiological change in the skeletal muscles continues or the muscles will contract. Now, this is the acetylcholine release and at the neuromuscular junction. What will happen? This is the postsynaptic neuron. This will release with the activation of calcium ions the so many molecules of acetylcholine, and at the postsynaptic membrane, here the acetylcholine receptors will receive these acetylcholine molecules, and this red part is the acetylcholine esterase. This enzyme dissolve or neutralize this excess of acetylcholine or break down in the acetate and the choline moieties. Okay. So, this is the complete physiological function during a complete skeletal muscle contraction. Thank you.